Hello students, in today's lecture of Principles of Inheritance and Variations, we will discuss three topics. Number one is the non-allelic interactions, then the polygenic inheritance, and lastly the sex determination in the honeybee, then in humans and in birds. First, we will discuss the non-allelic interactions. So, non-allelic interactions are those where the alleles of two or more independent genes interact to produce a phenotypic expression. So, this is basically exception of Mendel's law, because here we can see the blending of two alleles. Okay. Now, for example, we can see the epistasis. Epistasis means when one gene hides the effect of other gene. Effect of other gene. Okay. And we can see two types of epistasis mainly. One is the dominant, and other one is the recessive epistasis. Okay, and in the dominant epistasis, we can see the ratio in the F2 generation of dihybrid cross is 12:3:1. And in the recessive epistasis, we can see the ratio is 9:3:4. Similarly, in case of duplicate gene, the ratio is 15:1. Then, in case of inhibitory gene, the ratio is 13:3. In case of supplementary gene, the ratio is 9:3:4. And in case of complementary gene, the ratio is 9:7. In case of complementary gene, two different kind of allele, they complement each other. So here all of this ratio are exception from the ratio of Mendel's dihybrid cross. The phenotypic ratio of 9:3:3:1. So here all of these terms, they don't follow this phenotypic ratio, and they show different kind of phenotypic ratio like 12:3:1, 9:3:4, and so on. So this is all about the non-allelic interactions. Then we will discuss the polygenic inheritance. So in nature, we can see that the characteristics, they are spread across a gradient. Okay, they are not just two distinct characters like the Mendel experiment. So in this kind of characteristics, the characters are controlled by more than one gene. Okay, so this phenomena is known as polygenic inheritance. So, for example of this kind of characteristics is the human skin color, and also the heights of human. So these kind of characteristics are variable and they are spread across a gradient. Okay, so for example, suppose the human skin color is controlled by 3 genes. Those are A, B, C gene. Okay. And the dominant genes, for example, the capital A, then capital B, capital C. This is responsible for the darker skin color, and the recessive trait, or the small a, small b, and small c is responsible for the lighter skin color. Okay. So this kind of cross will produce 8 gametes, such as the capital A, capital B, capital C, then the capital A, capital B, small c, then capital A, small b, capital C, small a, capital B, capital C, and capital A, small b, small c, then small a, capital B, capital C, and small a, small b, capital C, lastly small a, small b, small c. So 8 gametes will be produced from these 2 genotypes, and these gametes in the F2 generation will produce different skin color individual in a gradient. So in the F2 generation, of the humans, the skin color will be in the phenotypic ratio of 1:6:15:20:15:6:1, and this will move from the lighter to the darker. Okay, so here in this image we can see this is the 1, then 6, here the 15, then this is the 20, then again the 15, then the 6 and 1. Okay, so here the combinations occurs in the gradient from the lighter to the darker. Okay, so this was all about the polygenic inheritance. Then today's last topic of discussion is the sex determination. So the sex determination was first observed by Henning in 1891 by his experiment where he observed specific nuclear structure present in the spermatogenesis. Okay, and in 50% of the structure, the nuclear structure was present, and in the other 50%, the nuclear structure was absent. Okay, and he termed the nuclear structure as X body. Okay, and after fertilization, he saw that the 50% individual with the X body, they produced the female offspring, and The other 50% without the nuclear structure of the X body, they produced the male offspring. Later, he named the X body as the X chromosome. X chromosome or the sex chromosome. So, the sex chromosome means the chromosome which is involved in the process of sex determination. Okay, and other chromosomes which are not involved in the process of sex determination are known as autosomes. So autosomes are chromosomes which are not involved in the process of sex determination. Okay. Then we will study the 3 types of sex determination. Those are the XO type, means the XX or XO type, and XY type means the XX, XY type, and the lastly the ZW type means the ZZ, ZW type. Now, the first of type, the X0 type or XO type, we can see this kind of sex determination in the earthworm and insects. Then, the XY type of sex determination is seen in the Drosophila or in the mammals, for example, the humans. Then we can see the ZW type of sex determination mainly in birds and reptiles. Okay. Firstly, we will discuss the XXXO type of sex determination, where the female individual is XX, or it is also known as the homomorphic. Means in the homomorphic organism, two morphologically identical chromosome are present. Okay. Then the male individual in this case is XO. Here the O stands for the null because here only one X chromosome is present. Okay. Now, the perfect example of this type of sex determination is the honeybees. Okay. So, in honeybees, we can see 2 types of females. One is the queen and other one is the worker. And in the male, we can see the drone type of bees. Okay. So here the female has the phenotype of XX and the male has X0. Okay. So in the process of the gametogenesis, the female individual is the XX. When this is crossed with the male individual of the genotype X0, here this will divide in the process of meiosis and the male will divide in the process of mitosis. Okay, so here This female one is with 36 chromosome, or the 2n diploid is present here. Okay, and here the n present, which is the haploid. Okay, and here the n is equals to 16, means the 2n is 32. Okay, now the result of meiosis will produce one n and the other n. Okay, from the female individual, means 16 and 16. Okay, and the result of mitosis in the male will produce one single N gamete with 16 chromosomes. Okay, because it was previously in haploid condition. Then the fertilization of these two will produce the 2N here with the 32 chromosome, and that will be the female with the XX genotype. And here the not fertilized one will produce the male individual with the X0 genotype. Okay, so this was all about the sex determination of the honeybees. Okay, then we will discuss the XX and XY type of sex determination, where the female individuals have two X chromosomes and they are denoted as XX, and the male individual has one X chromosome and one Y chromosome, and they are denoted as XY. Okay, so here for the male individual, the Y chromosome is must. So we can write it as Y chromosome is essential for male individual. Okay, so here if the father is XY and the mother is XX, so their generation will produce 50% son and 50% daughter. Now, how this calculation will occur? So here, if this is the gamete produced by the father, the X and Y, and here the gametes produced by the mother. So the mother will produce only one gamete, which is the X. Okay, so then this cross will form XX, then XY, here again XX and XY. So here these two XY and XY genotype will produce male individual, and these two with the XX and XX genotype will produce the female individual. Okay, so here the ratio of male and female in the offspring will be 1:1. Okay, so this was all about the XX and XY type of sex determination. Then we will discuss the ZZ and the ZW type, where the male individual is denoted as ZZ or the homomorphic. Okay, and the female individual is denoted as ZW or the heteromorphic, which is exactly opposite of the XXXY type, because in the XXXY type, the male individual was heteromorphic and the female individuals were homomorphic. Here, in the ZZ and ZW is just the opposite. Okay, and we can see this kind of sex determination in the birds and the reptiles. Okay, so here this kind of sex determination will also produce 50% male and 50% female. So here the male individual will produce two gametes of one type. Okay, so the male individual will produce the Z type of gamete and the female individual will produce the Z and W as gamete. Okay, so here the Z and W, and the offsprings will be ZZ, again ZZ, ZW and ZW. So here this ZZ and ZZ of the first row will be male, so 50%, and the ZW and ZW, this will be female of 50%. Okay, so this was all about the ZZZW type, which is all about the sex determination. Okay, and the end of the part 4 and the genetics. In our next class, we will discuss the pedigree analysis. Pedigree analysis. Okay, in our next class.
To learn difficult concept of physics, chemistry and biology within few minutes to crack NEET and JEE with fun, like, share and subscribe our new YouTube channel ShantU. You will get conceptual videos, highly effective notes, short tricks, mock tests and much more. Hope we shall meet there.